So thanks very much for this very nice introduction. I also have to say I enjoy this meeting a lot. There's a lot of young energy, a lot of enthusiasm, so there's hope for the future. So today I'm going to show you some of our more recent work on what we think brain oscillations, in particular in the alpha and gamma band, are doing for human cognition. And what I'm first going to do is to tell you about the power of these oscillations, the magnitude, and how that activity might be involved in routing of information between different nodes in the brain network. And then I will switch to tell you some of the ideas and work we have on the phase of the oscillations. So first, the power story, so to speak. So over the years, we have been working with this notion of trying to uncover what is the dynamical mechanism supporting routing between different regions in the brain network. And what you have to think about is that in a split second, different regions have to communicate in order to support a given task, whereas other regions do not have to be involved. So how does the brain achieve this? There are several ideas, some ideas being maybe synchronization between different nodes in the network. However, what we have been pushing forward is this notion that could it be that it is basically the suppression of nodes not important for a given task that sets up the brain work, in a sense that if all the nodes not required for the task are suppressed, then it allows the different nodes that are not suppressed to engage, communicate, and then do the necessary computation. So that's part one. And part two is that we think we have now good evidence that this shutting down of the different nodes is reflected by alpha band activity. This seems to occur in the visual system and somatosensory system, but not only that, we also see this alpha activity associated with task disengagement in higher cognitive regions. For instance, prefrontal cortex, left prefrontal cortex, when you engage the language network, is associated with a decrease in the alpha band. So the story here we are promoting is sort of derived from what we have learned from the sensory regions, but we think that it generalizes to other brain regions as well. And of course, what is also very important is what sort of frontal regions are involved in doing the top-down control of the, say, posterior regions in which the alpha oscillations are being modulated. So we have also been doing some work in trying to uncover these control regions being involved. So I should also say that this view that we are pushing on the alpha activity as being an inhibitory is in somehow contrast to the idling notion of the alpha band activity. So you're probably all familiar with the notion that if you sit there, close your eyes, do nothing, the alpha activity shoots up. So it used to be such that people thought that the alpha activity just reflects a state of relaxation or people not doing something. So we have taken issue with that point, and we now strongly believe that you have to refine that view in order to think about the alpha activity as doing the active suppression of regions not involved in a given network. So in order to address this, we have been using MEG as our core tool, but also, as you learned from Marcus's talk yesterday, one tool is not enough these days because every tool has limitations. So we have also been combining MEG with other techniques like structural MRI, brain stimulation, and so forth. And I'll also show you some data where we have been trying to link structural measures from MRI to our MEG data. I should also mention we have just been setting up a new MEG system at the Center for Human Brain Health in Birmingham. So it's a new brain imaging center focusing on cognition and human brain health. And we're quite happy that we finally got the MEG up running here in January. So now we are setting up to go full speed on our MEG research there. So I'll start by showing you some data from a spatial attention task, both to underscore how we work with MEG data, but also some of the newer analysis we have been doing in order to uncover the control, top-down control of brain oscillations. So the paradigm you see here is somewhat complicated. So there's a cue being presented first, and this cue here just indicate people that they have to attend to the right, and then to the location here at the right, and then a few seconds later there's a target array coming on. So this is a fairly complicated task, but all what you should think about is that there are some instructions asking the subject to attend to a given location. Now we just sort the trials according to left versus right attention, just to keep things simple, right? So when we do this covert attention to the left and the right, what you typically see with EEG and MEG data is this hemispheric lateralization. So the time frequency representations you see here on the top right plot indicates the power as a function of time, and blue indicates that the alpha power contra-lateral to the cue is being depressed, right? So if subjects are asked to attend to the left, the alpha power over the right hemisphere goes down, but over the ipsilateral left hemisphere, alpha activity goes up, and that's indicated by the red color in time frequency representation. And we can also look at the topography, and there you see this very clear lateralization. So this kind of left-right attention paradigms is sort of the workhorse of a lot of the studies we are doing, because you have the top-down control there, and you also have the regional specific engagement and disengagement of in this case, the visual regions. Furthermore, since we can do MEG, we can localize this depression of or increase of the alpha oscillations to the early visual regions. So no big surprises here. There's a lot of studies showing this. However, in this study, what we then asked is what sort of regions are involved in doing the top-down modulation of the alpha band activity. In order to do this, we have been using the measure of Granger causality, and you might be familiar with that measure. So basically, you can look at two time series, and then if you can then ask if one sort of is in charge of signal A is in charge of driving signal B, or the other way around. You can also do this in the frequency domain, so you can ask what sort of regions are involved, in this case, in driving the oscillations in the visual cortex. And when we apply this measure, we get a spectra as you see to the right. So what you have to think about is that we identify a target region, meaning visual cortex, and then we go in and ask, in this case, if the frontal eye field, so that's what you see on the MRI plot, the red blob there, that's the right frontal eye field. So now we have calculated the Granger causality between the frontal eye field and the posterior regions, and what you should notice then is that there's the blue line, and that's indicating the top-down, meaning frontal eye field to visual cortex. The red line is indicating visual cortex to frontal eye field. So what you should appreciate here is that it's selectively in the alpha band that there is a top-down drive from frontal eye field to visual cortex. So now you might ask, why just frontal eye field? Could there be other regions involved? And indeed, that is most likely the case, right? But what we have been doing here is that we have been using visual cortex as a reference region, and then we have been exploring all other, say, grid points of voxels within the brain. And when subjecting this to a fairly conservative multiple comparison correction, then it's only the right frontal eye field we find being involved in doing the top-down control of the posterior alpha activity. So, of course, this does not mean that it's only the frontal eye field, but the frontal eye field is sort of the tip of the iceberg, right? So there's also other regions involved. And again, as you heard from Marcus's talk yesterday, the basal ganglia also play a strong role in various kind of cognitive control. And that's something we also have become interested in more recently. So there's the well-known connectivity between prefrontal cortex and the basal ganglia or the striatum, right? So people talk about the fronto-striatal control network. However, when you're using MEG or EEG, it's very difficult to get to the subcortical regions like the basal ganglia and striatum. So what do we do there? Well, I'll show you some data where we have been relating structural MRI to MEG data in order to get a take on what these subcortical regions might be doing. And in this case, we have again been using a paradigm on hemispheric lateralization with an extra twist. And this extra twist is that the items we are using were either associated with reward or losses, right? So before doing the actual task, we trained subjects on different Chinese characters in this case. And some of those were associated with a monetary reward, and some of these became associated with a loss. Then we used these characters in a lateralization paradigm again, where people are cued to the left or the right, and then the items they were tending to were either associated with reward, loss, or they were neutral items, right? So we then asked to what extent the posterior alpha oscillations were modulated by these reward contingencies. And on the top plot, you just see the sort of normal alpha lateralization when looking at the alpha activity for attention left versus right, right? What you should appreciate in the bars, in the lower bars, is that if the target was associated with either reward or loss, there was an increase in the alpha lateralization. So this is not that profound, right? It most likely means that if you are attending to an item and you know that that is associated with reward, it's going to pull your attention even more, right? So you are attending more to items associated with reward. And the same holds for losses, right? So if you attend to an item associated with a loss, there's also a stronger pull in attention there. Okay, so that was somehow the prelude. So we have a paradigm where we can modulate the modulate the alpha oscillations in visual cortex. Furthermore, there's reward contingencies sort of boosting this modulation. So, as I mentioned, we are interested in the striatum or the extended striatum, the basal ganglia structures. And in order to look into those, we have been using function MRI and then an algorithm from SFL called FIRST. And what that algorithm does is that it can automatically identify the different basal ganglia. So the color codes here indicate the globus pallidus and the hippocampus, the caudate, and the putamen, right? So these regions have all been identified automatically, and we can do that in individual subjects. So now we are employing a little trick. And what we are doing is that we are looking at the hemispheric lateralization in terms of the magnitude of these structures, right? So it could be that some subjects have a bigger left than right caudate and so forth. So we have been quantifying this. And in the bar plots below, you see a histogram of these lateralization indices, right? So what you should appreciate is that, for instance, when it comes to the globus pallidus, there's a left lateralization. But when it comes to, in this case, difficult to see, the nucleus accumbens, there's a right lateralization over subjects, right? But it just means that some subjects have, typically subjects have a stronger right than left nucleus accumbens. So what these inherent lateralizations means, we don't really know yet, but it's actually quite striking. We have been able to reproduce this in different studies, so it seems to be a robust phenomenon. Anyways, we have been using this lateralization now to ask, could it be that subjects with a larger left than right globus pallidus also can modulate their left versus right hemisphere alpha stronger, right? So to do this, we have also derived an index for this ability to modulate your left versus right alpha band activity. So don't think about pluses and minuses here, just think about that we have a measure for how much the alpha activity goes up and down in the right hemisphere as compared to the alpha activity going up and down in your left hemisphere when attending left and right, right? So now we can relate that hemispheric lateralization in ability to modulate the oscillations to the structural lateralization in terms of the subregions of the stratum. And when we do this, we find quite a robust correlation for the globus pallidus, right? So if you consider one of these subjects up here to the right, one of these subjects would have a larger right globus pallidus than left. They can also modulate their right hemisphere alpha stronger than their left hemisphere alpha, right? So this is a bit naive, right? So we just say like a bigger subcortical brain structure, a larger subcortical brain structure is associated with a better ability to modulate the oscillations in the same hemisphere. So it's a bit naive, but it actually works. And this is also something we have been able to reproduce recently. So far so good. So we have now, I've just shown you the data here for the globus pallidus, but it turns out that if we do a GLM analysis taking these different subregions, the 4 subregions of the extended stratum into account, it's only the globus pallidus for which we see this relation being robust, right? So to make a long story short, we think that we can, we think that the globus pallidus somehow is involved in doing the top-down control of posterior alpha oscillations. I talked about the reward contingencies. So not to make things too complicated, but here we can think about 2 reward contingencies. It could be reward left or right, or just 1 reward contingencies. It could be reward just to the right and then neutral to the neutral reward contingency to the left, or it could be neutral, right? So, but it turns out that the more reward contingencies involved in your task, the more robust this correlation between structure of the globus pallidus and the posterior alpha oscillations. So this here also suggests that it is in particular in reward type of tasks that the extended stratum kicks in doing the top-down control, right? Which is, of course, consistent with what we know about the basal ganglia or the stratum as playing a role not only in motor tasks but also in reward type of tasks. To make a long story short, so we are in the process of trying to uncover the fronto-striatal network involved in top-down control. So what we have to think about is not only the structures involved, also the pathways and also the mechanisms by which this control is exercised, right? So far we've just been scratching the surface of this, so there's a lot of work ahead in the future to further uncover these type of networks and mechanisms. So why do we do this? Well, first of all, we are of course interested in just the basic mechanisms, but we're also interested in what's going on in individuals that have problems in allocating their attention, right? So, for instance, the elderly and also patients with ADHD have problems in modulating attention. We have also been able to demonstrate that these groups have a reduced ability to modulate their posterior alpha oscillations. So we are trying to make use of these findings in order to also identify the fronto-striatal network and the mechanisms there that might be malfunctioning in these patient groups, such that we eventually can do a better diagnosis and intervention, right? So there's also a translational element to this. Since these patient groups have an inability to modulate their posterior oscillations, but we don't think it's because there's something going wrong in the sensory regions producing these oscillations. It's probably the fronto-striatal regions where the problems are. However, we can use the posterior oscillations sort of to pull in a network in order to identify these fronto-striatal regions and then try to figure out what's going wrong. Okay, so there was some of our work on how we address posterior alpha oscillations in terms of their magnitude and power. But what we also want to understand better is the phasic modulation exercised by these oscillations, right? So, of course, if you are interested in oscillations, you need to embrace what these oscillations are doing in a phasic sense. Otherwise, it just doesn't make sense to have this interest in oscillations. And what we are really trying to do is to identify the mechanisms that these oscillations might be involved in implementing in terms of coordinating neuronal activity. So the first study I'll show you is actually not a human study, but it's data from monkeys. So what this here is a study on Laminar data recorded by Axel Meyer, back then working with David Leopold at the NIH. And what they have been doing is to implant these laminar electrodes in early visual regions in V1, right? So these laminar recordings is still a kind of EEG recording, right? It's just like you have a lot of finely spaced electrodes on a little pen, if you will, that you penetrate through the cortical layers. So it allows you to infer what's going on in a different cortical laminae. And if you look at this plot, you can almost, with the naked eye, see that in the deeper layers, there's some sort of alpha activity going on, whereas in the more superficial layers, there's faster gamma band activity. So we're interested in what is the relationship between the alpha and the gamma band activity. So what we did here was simply to look at the data from the different layers, or the deeper layers, and then cut out snippet of those data. And then we phase align those traces according to the peak of the alpha band activity. Then for each of these traces, we calculated the time frequency representation of power, and then we averaged all this together, right? So it should probably be no surprise that for the deeper layers, we then see this like blip of alpha activity, because that's how we analyze the data, right? But what you should appreciate here is that when you look at the time frequency representation of power, then at higher frequencies, there's a clear modulation by alpha phase, right? So this here suggests that you have ongoing alpha oscillations that are actively modulating the faster gamma band activity. And you can think about the gamma band activity as supporting the actual computation, but that computation is clocked in a phasic way by the alpha band oscillations. Furthermore, what we also found interesting here is that the alpha activity was strongest in the deeper layers, and the gamma band activity was strongest in the more superficial layers. And you might be aware by the notion that what we measure with EEG and MEG is reflecting the dendritic activity of the layer 5 and 6 cells. So these layer 5 cells have strong dendrites going through all the cortical laminae, right? So with that consideration in mind, we are actually sort of biased to dendritic currents projecting to cells in the deeper layers. So this might actually explain why we see the alpha activity so strong in humans when recording with EEG and MEG, because when you talk to monkey electrophysiologists, they don't see the alpha activity as strong. But that's probably because they're typically recording in more superficial layers, right? So this notion that alpha is in deep layers and gamma in the more superficial layers sort of can help to resolve that conundrum that we see that we are probably biased using EEG and MEG recordings to see the alpha activity being extra strong. We can also do these more sophisticated phase-to-power analysis. So you might not be familiar with the analysis, but the idea is that we can explore all phases of the slow oscillations and all we can explore the phases of the slow oscillations in a broad frequency range, and the power of the fast oscillation and broad frequency range. And when we do this, what sort of pops out as being the most salient is the alpha phase to gamma power coupling. So there's also some consideration on current source densities, but let's not go into this in the interest of time. I should also say that we could also do a correlation between the length of the bursts of the gamma band activity and the alpha activity. And when we do that correlation, we see an inverse relationship. So this is again consistent with the alpha activity being inhibitory. So the stronger the alpha band activity, the shorter the bursts of the gamma band activity. And it's going to be important for the next study I'm going to tell you about. Before getting to that, I should say that we are also playing with some ideas of thinking about the canonical microcircuits. So you might be familiar with the notion that the laminar organization that you see in visual cortex seems to be somehow ubiquitous over cortex, right? So there's something special about how these cortical layers are organized. Now it seems like this organization of the layers is also paralleled by the dynamics. So what we now can start to think about is not only sort of canonical microcircuits, but also canonical dynamics, if you will, in the cortical layers. And we have some speculations about feed forward and feedback, how that matches to the neuronal dynamics, but also how it matches the microstructure that you see in the cortical layers. So I'm going to switch gear a bit and point you to some data from the rat hippocampus. I'll do this and then after that loop back to alpha oscillations in humans. But this is a study going back to John O'Keefe, and you're probably all aware that John O'Keefe got a Nobel Prize for discovering place cells in rats. So not only did he do that, he also has been investigating the relationship between these place cells and ongoing oscillations. So the plot here you see is an example where in the lower trace you see the ongoing theta band oscillations in the rat. And I should say that the theta band oscillations in rats are defined all the way from down to 5 but up to 12 hertz, right? So there's an overlap in what people think about theta oscillations. There's an overlap in the frequency range, at least between theta oscillations in the rat and then alpha oscillations in humans. So that's also going to be important for later. But nevertheless, what you then see, the long vertical lines is just the theta oscillations being sort of segmented. And the small dots you see there indicates the firing of individual place cells. So what you now should appreciate is that as the rat enters the place field, and the place field is defined by the firing of the spiking as you see there, right? So as the rat moves through the place field, first you see the firing being late in the theta cycle, and then the firing goes to earlier and earlier and earlier phases of the theta cycle. So when the rat is leaving the place field, the firing is quite early in the theta cycle. So what does that mean? It means that if you were to sort of read the rat's mind and decode his position, well, then you get some information by looking at the firing alone in terms of firing rate. But if you take the theta phase into account, you can do an even better job at reconstructing the rat's position, right? So what this here means is that there is some sort of phase code going on where the phase of firing actually carries information. And this notion that the firing within an oscillatory cycle being important is something that we have been playing with over the years. So I will point you to a study now that we are or have been working on with John Lisman, in which we think this phase-specific firing is important for working memory maintenance. And this study, so sadly John Lisman passed away last year. Nevertheless, what he has been pushing over the years is this notion of phase coding during working memory maintenance. So this goes back to a paper he put forward with Idiot in Science in 1995. And what Lisman and Idiot proposed was that you have these ongoing oscillations, and then you have individual working memory representations riding on these oscillations, right? And furthermore, these individual items are segmented by the gamma band oscillations. So the elements you have here is the slow oscillations. They thought about this as being theta oscillations, and on top of these theta oscillations, you have the individual items riding. They are then clocked by the gamma band oscillations. So now we have, again, this phase to power coupling between, in this case, theta and gamma oscillations. Furthermore, the number of gamma oscillations, according to this proposal, is determining how many items you can keep in your working memory. Furthermore, the frequency of the gamma oscillations is going to determine how fast you can retrieve from your working memory. So when looking at the numbers, meaning the gamma oscillations per theta oscillations, you actually can match this with the capacity of your working memory being maybe about 5 to 7 items. If you're familiar with the Sternberg paradigm, then that measures how fast you can retrieve from working memory. There, that retrieval time also fits with the gamma oscillations. So this is a bit of a crazy proposal, but there is, in the sense that there's, when you look at the psychophysics, then there's some consistency between the brain oscillations, the frequencies, and how you operate in terms of behavior when it comes to working memory maintenance and retrieval. Nevertheless, it has been somehow difficult to find support for this framework, but we think we now have some data providing quite compelling evidence for this scheme. So in principle, the scheme is quite testable, right? You should be able to see this phase-specific activation of the individual items. However, that has been quite difficult to find evidence for this, because how do you get this phase-specific information in humans? Nevertheless, we have some ECOG data here from Josh Jacobs that has made this possible. And what Josh has been able to demonstrate is that there are some electrodes, when looking at ECOG data in the human working brain, there are some electrodes that respond selectively to different kind of letters, right? So you might have one electrode, as you see here, that really responds to Q. So when Q is being presented, you see this broadband gamma activity spanning maybe from 80 hertz and up, as you see in the plot there, right? When you present another letter, you don't see this gamma band activity. So this by itself is somehow quite incredible, right? So at least before seeing these data, I always thought about this. In this case, this letter specific representations were reflected by distributed neuronal firing, right? But in fact, it turns out that there are electrodes, these ECOG electrodes, that respond quite selectively to specific letters, right? So this is good news when testing the working memory model, because now we can look at the representational specific activation. So we can look at this broadband theta activity in relationship to the phase of the ongoing theta alpha oscillations. So this is what the data in general looks like when we are presenting the working memory stimuli, right? So these are time frequency representations of the ongoing activity. And what you should appreciate here is that each time we present a letter, there is this blip of broadband gamma band activity, and then there's suppression of the alpha band activity. What you then see during the maintenance, so that's the more extended time frequency plot, is that there's an increase of the alpha band activity, and the gamma band activity is no longer strong. And then the last plot is the presentation of the probe item, right? So in this case, three different letters were presented, Q, D, R, and then in the end Q comes on and subject you have to say yes or no to whether Q was in the list, right? So each time we present these items, there's a burst of gamma band activity to suppression of the alpha band activity. During the maintenance, there's the sustained alpha band activity. You can also see here we sorted the plots according to letter selective versus we sorted the plots according to whether the electrode responded to an item or not, right? And then we see this modulation. So when looking at the data, the first observation is that during the maintenance interval, we see a strong phase to gamma band, coupling between alpha phase and gamma power, right? So this by itself is a bit surprising because we don't see too much gamma band activity during maintenance, but when we look at the gamma band activity with respect to alpha phase, we actually see a modulation, quite a strong and robust modulation of gamma power with respect to alpha phase, right? So when we think about the ingredients from Lisman's model, first the coupling between the phase of slow oscillations and then the gamma power, then we indeed see that in these data. But of course we wanted to go further than that, and we wanted to look at this phase-specific activation. So this plot here becomes a bit complicated, but what you now should appreciate is that we record as an example from an electrode that responds to Q, right? So when Q is the first item in a list, what you then see in an example trace is that Q should be activating early in the theta alpha cycle, right? So that's identified by this little gamma blip that you see in the first example trace. However, if you now presented Q as the second item in the list, you should see the gamma blip in the middle of the theta alpha cycle, and if Q was the last item in the list, you will see the gamma blip in the end of the theta alpha cycle, right? So since we have 10 minutes left. So since we have this representational specific activation as measured in the ECOG electrodes, then we can actually look into this scheme. And indeed, we find good support for this phase-specific activation, right? So again, a bit of a complicated plot, but again, imagine that you are recording from an electrode that responds to Q. The red line is then when Q was the first item in the list, right? So, and what you have on the x-axis is the phase of the theta alpha oscillations. And what you then see when the Q is on the first position in the list, you see the gamma activity being early. The green plot is when Q was the second item in the list, and the blue plot is when Q was in the end of the list, right? So you'd see this phase-specific gamma band activity with respect to position. And now I just gave one example for Q, but of course we did this for all the letters and the different combinations and so forth. So this turns out to be quite a robust effect. So finally, after a good 20 years, we now think we have some support for Lisman's initial notion that different items are being maintained during working memory maintenance by phase-specific firing. So I don't have too much time left, so I was going to show you some data on saccades. So I'll just briefly walk you through this, because it shows one of the newer directions we are going to take as well. So it's a bit of a side issue, but it sort of links up to the rest of the story. And the idea is that, of course, when we study visual attention, often we avoid saccades because saccades creates artifact, right? But nevertheless, what we do during daily life is to saccade 3 to 4 times per second, right? So if we actually are ignoring saccades, we are missing an important part of what we are doing when exploring the visual scene. Another issue with saccades is that it's not obvious how they link to ongoing brain oscillations, but what we think now is that we have good evidence that saccades are actually locked to the phase of ongoing alpha oscillations. And we think that is becoming quite important because these alpha oscillations are not only controlling visual processing, but also how the motor system, in this case controlling saccades, are sort of linking up to timing the whole network. And simply what we did was to ask people to attend to different pictures being presented, but we, in this case, allowed subjects just to saccade as much as they wanted to do to each item. And what we then did was to do a phase analysis. What you should appreciate here is at time 0 you have the saccade, and then we look at the brain activity just before the saccade. And these here are actually intracranial data recorded from patients that have these depth electrodes into visual and more deeper regions in parietal and temporal cortex. And the color code here represents a phase alignment, right? So what this here actually means is that the individual trials are phase aligned prior to the saccade, or to put it differently, each time you saccade, your saccade is locked to the phase of the ongoing alpha oscillations, right? But what you also appreciate here, this here is actually a contrast between items that you later remembered and later forgotten, right? So it's in particular when you're able to remember the items presented on the screen that your saccades become locked to the phase of these ongoing alpha oscillations. And I should say this, what I showed you before was intracranial data. We see the same in human MEG data. So this turns out to be quite a robust effect. So I'm saying this because what we think actually is going on is that you have ongoing information. This information is somehow clocked by the phase of the ongoing alpha oscillations. Furthermore, we also think about locking between the activity or the phases of alpha oscillations in different networks. In this case, it would be visual cortex to the hippocampus. This locking is then reflected in the information transfer. And furthermore, the saccades themselves also locks on to the phase of these oscillations that are coupled between different regions. So what I said here last is a gross overstatement compared to the data we have, but that's sort of the bigger picture we are working towards. So I'm just going to say a few words about the future directions we are going to take, right? So we have been talking about the sort of theta alpha oscillations in the rat hippocampus and also in humans doing working memory maintenance, and how the phase of firing is important for representing information. We think that this might also be important for alpha oscillation in the visual system. And again, this pertains to how you process a visual scene. So in this case, you might be on your bike going to work. You have visual information coming in, a lot of visual information that needs to be prioritized. So you do need to focus and attend to the lady on the orange bike, but that's not sufficient for being able to navigate here, right? You also need to allocate some resources to entertain other items you might have to attend to in a split second later. That could be the guy on the yellow bike and maybe the traffic light. And then on top of that, there's a lot of irrelevant information coming in. So how is the brain doing this? So to make a long story short, what we think is that the alpha oscillations serve to segment the visual scene such that you have competing visual input. And as we talked about before, the alpha oscillations are inhibitory, but we think about them as being pulses of inhibition. So at the peak of inhibition, all information is blocked, all visual information. But as the inhibition ramps down, you get the activation of the most excitable visual item. That could be the lady on the orange bike. And then as inhibition ramps down a bit further, then it would be the guy on the yellow bike and then the traffic light, right? So what we have been doing here is actually stealing some ideas from theoretical work on the rat hippocampus and then applied those ideas to the human alpha oscillations. And we think that these oscillations are inhibitory, but this inhibition sort of creates a filter, and as inhibition ramps down, you get a sequential activation of competing representations. So you get a little to-do list, if you will, that then can be processed one at a time. So you get a little pipeline, if you will, that allows you to do a sequential computation of items being presented in parallel. So we can have a more broad discussions about the motivations for doing that. We can also discuss what sets the excitability of the individual items and so forth. Anyways, this is the bigger scheme that we are working towards. We have several ideas for how to pursue this. So even though this scheme might be somehow speculative, so take it with a grain of salt, it's also fully testable, right? So all what we need to do, so to speak, is to find evidence for this phase-specific activation along the phase of the alpha oscillations. But this is work for the future. So I will end here by summarizing. We now believe that these alpha oscillations are inhibitory, so they somehow limit the processing, but they don't completely abolish the processing. It's more that they inhibits in a pulse sense, yet there is still the possibility for phase-specific activation. So we think about very strong alpha oscillations creating small windows of opportunities, but if the alpha activity is diminished, you have a longer duty cycle, a longer windows of opportunity where you then can have this phase-specific code. And this is the bigger scheme that we are trying to uncover. Furthermore, as I mentioned in the start of the talk, we're also trying to identify the interactions between frontal control regions and subcortical regions and how they're involved in modulating the posterior alpha oscillations. So in interest of time, I will end here and acknowledge some of the people and collaborators being involved in doing this work. And thanks very much for your attention.
Well, thank you very much for this lovely talk full of new data and new information. Is there any question to Professor Ole Jensen?
Concerning the first study that you showed, the one with communication from frontal eye field to visual cortices, I was wondering about the interpretation of the Granger causality there.
Yeah.
I mean, the interpretation, the obvious interpretation is that frontal eye field controls the alpha amplitude, for example.
Yeah.
But I was wondering whether another interpretation, kind of similar, would be possible, that the alpha is already present in the visual cortices, and because it creates those windows of these duty cycles, then any influence from frontal eye field is confined to those duty cycles, so it's cyclic in that manner. Would it be possible, or is it a valid interpretation of this effect?
Yeah. So I mean, so first there's a clarification. So the analysis does not allow us to state that the frontal eye field is controlling the power of the alpha oscillations, but it tells us that there's a phasic control of the posterior oscillations, right? So I don't know if that helps to address your concern.
So in general, both interpretations would not be distinguishable in this case, because if there is cyclic control from frontal eye field that may influence the amplitude of alpha in the visual cortex, right?
Yeah.
But it may also not influence the alpha amplitude, but just be confined to those duty cycles.
Yeah, I think it's the latter we have support for, right? So it is actually the phasic control. It's not the magnitude of the oscillations.
Okay.
But it's good you point this out, because it of course is an important difference.
Right. So thank you for clarification.
Yeah.
Thank you for a very nice talk. I was wondering about why didn't the thalamus show up? Because in the older models, the Skinner and Yingling model, etc., it's always the thalamocortical loops that are proposed as the explanation for the alpha. Why does it not show up in your analysis?
Yeah. So I'm very sympathetic to the view that the thalamus is highly involved in doing the control of these oscillations. The only thing is with MEG and EEG, we cannot detect reliable thalamic activity. But there's older work by Lopez da Silva showing that the pulvinar and also LGN is phase coherent with alpha oscillations. This was in dogs. Sabine Kastner has a paper where they show pulvinar to extrastriate visual cortex. So definitely I think thalamus is highly involved. Also what we showed about the globus pallidus, indicating that globus pallidus is involved. There's two major routes that globus pallidus can project back. So that one would be to an anterior thalamus, and then prefrontal cortex, and then down to posterior regions via the superior longitudinal fasciculus. But there's also the possibility that the globus pallidus projects to an anterior thalamus, that we know it does for certain, and then you have anterior thalamus communicating to pulvinar, and then pulvinar to visual cortex. So these are things we are entertaining, but I think we probably need animal studies to uncover this.
I have one last quick question, since I think there aren't any more. Since MEG is very expensive and rare equipment, would we be able to replicate your findings using EEG?
To some extent, but I would like to state that, for instance, alpha oscillations, since people used to record those with EEG and with few electrodes, there was the notion that alpha reflects the state of the brain, right? The fact that we are using MEG allows us to get to the more regional specificity, and there has really been sort of a big shift in how we understand oscillations. And the main shift is to understand these alpha oscillations as not reflecting brain states, but say states of individual nodes in a network. For that, you need the local specificity, and you can do that better with MEG than EEG. But that being said, some of the stuff we are doing is also quite feasible with EEG, right? So it's just you have to be more careful when it comes to where the activity is being generated. But there's good news ahead, and that is, you mentioned that MEG is very expensive, but it will get cheaper because there's a new sensor development. They're called optically pumped magnetometers. So it's a new kind of optical type of magnetic sensor that works as good, if not even better, than the conventional squid sensors. So a few weeks ago, there was a Nature paper out demonstrating that these sensors can be used for human MEG. Of course, there's maybe 5-10 years ahead of development, but there's news ahead for MEG research. It will get cheaper in the future because we don't then have to rely on the liquid helium and so forth. So, but until then, if you only have an EEG system, of course, by all means, go ahead. You can do a lot of great research with that. Yet, I would still state that when it comes to localization, you have an advantage with the MEG.
Okay, thank you, and I'm happy that it's getting cheaper. So, thank you one more time very much, Professor. And I invite you to our seminars.